Well, thank you very, very much, Rebecca. Thank you very, very much for this introduction that took me through the ages. It's sobering, and thank you for the invitation. And I will hope to present a talk that will create the kind of path to interesting talks in the future. If I bomb, you'll never have a talk again. So I hope it'll go well. So I think I need to give just a moment's explanation of the title. So here is what I meant by the title. It is the presentation will aim to illustrate a path of real partnership between forms of intelligence that are inherent in the enormous emerging capabilities of AI and machine learning tools, and inherent in the acquisition of a deep understanding of biomedical systems with physics-based and biology-driven methodologies, which is what we have done for many decades. The context of this illustration is a specific investigation of something that many members of the society do themselves, the functional mechanisms of vital molecular systems. And the context is to highlight the advantage of this partnership that offers the depth of understanding and its use in design. So, given the extraordinary achievements and the even more extraordinary PR of the AI/ML developments, it's a good question why partner and just not use undiluted AI, because for one thing, it is free and available to all, and it does have interesting performance. One reason is illustrated, for example, by the most recent achievement. So here's a paper from 2025 entitled Molecular Analysis and Design Using Generative Artificial Intelligence, which is enormously powerful in creating new circumstances for understanding with multi-agent modeling. And I want to tell you, I'll emphasize several things from the paper. You see here, the model used features 7 billion parameters. And yet, from a chemoinformatics point of view, if you want, from a physicist and chemist point of view, it targets the properties of the molecules that are by using a principal component analysis of molecular properties. And the result that is presented from this enormous achievement is that we validated that, as predicted, increased dipole moment and polarizability is indeed achieved in the designed molecules. Now, I don't know about others, but to me, this was rather a disappointment, going through 7 billion parameters and achieving increased dipole moment selection. So it seems that if we do what we do, we are on our own for the moment and taking advantage only of understanding that has evolved from the scientific base of physics, chemistry, biology, and molecular modeling. Perhaps. The answer is no, we're not on our own. There are pioneers and giants in this field, and George Church at Harvard is one of them, who are really driving this field of AI-driven design. It happens to be that they talk here about protein design in September this year. And as you will see, the AI learning paradigms and model architectures that have come from it are enormous, and it would be right out silly not to take advantage of them as quickly and as thoroughly as possible. So we're not alone. More importantly, there are model architectures for questions that one asks and spaces that one visits that many of us already use and don't even think of them as advances in artificial intelligence. They're there, we know them, we use them. So there is hope. And that is, I think, in a very honest manner, what George writes in the Outlook. They say AI has advanced protein design from optimizing antibodies, which was a simple task with these simple tools that you see above. However, designing complex, multifunctional proteins, such as large multi-domain assemblies, or systems that we are very familiar with in our work, with intricate allosteric networks, remains challenging. And it highlights, of course, opportunities for future innovation. So this is the wedge, I think, that we can enter that we can use to create the partnership. And this is why we need to work on the intelligence partnership, the partnership between the two types of intelligence that I spoke about. I don't know what happened here. Sorry. And so what I would like to do is present in the context of these questions why we need to work with this intelligence partnership. So the context is a molecule that many of us have looked at and tried to understand and manipulate. And that is a transporter of cholesterol in the cell. This type of transporter, unlike the SSRIs and dopamine transporter and lipid transporters that we know, functions as actually, first of all, it functions intracellularly, and it functions as a shuttle service in the sense that this STAR-D4 extracts from membrane components in a target membrane that it approaches something, in this case, cholesterol, that it takes over to another membrane and deposits it there. So this is a type of non-vesicular transport that is essential to the cell. Why is it needed? Because of the differential distribution of cholesterol in the cell. As many of you know, cholesterol is very unevenly distributed. You see it go down here. It goes from high to low, and I will try to make sure I have a good colored here. As you can see here, it goes from high to low, and you can tell that the distribution is uneven, and that unevenness needs to be maintained because it involves active differential particles of the components of the cell, and therefore the differential distribution needs to be maintained and involves therefore active trafficking. And STAR-D4, this tiny protein, contributes a third of all the non-vesicular transport between the plasma membrane, the ER, and the ERC, which are extremely important. And our colleagues at Cornell have obtained extremely interesting information about what STAR-D4 does. And In a schematic manner, I will show you that it consists of one single soluble star D domain. And the N-terminal region is the part that embeds in membranes. And it is anchored to these membranes by the C-terminus of what will become familiar to you, I hope, which is helix 4, and another part of the molecule, the beta sheets, beta 1 and beta 2. This will play an important role in our analysis. And this embeds in the membrane in order to bring cholesterol cargo through the flexible gate, or to prepare the already loaded star D4 for sinking the cholesterol into the membrane, releasing the cholesterol into the membrane. So the experiments that were done on vesicles for sterile uptake and delivery, donor vesicles and acceptor vesicles, was done with fluorescence methods, which are well described and I'm not going to go into, but you just see that the sterile, whichever it is, when it comes to the fluorescent agent in the membrane, it changes the fluorescence. And they tested the vesicles with a variety of negatively charged components, which they understood to be the ones that are important for recognition in this trafficking. And I don't need to remind you, but I will do for completion. These negatively charged components of the membrane, although they're both PIP2 subtypes, they differ from a structural chemist point of view. They differ significantly because you see the distance between the important negatively charged groups is very different in the two. And nature has used this as a recognition sign. And you can see, I'm not going to go into details of this, but you can see that the ability to extract and deliver depends on the kind of PIP2 subtype that is present in the membrane. So the main conclusion from the experiments is that the efficacy of cholesterol trafficking, both extraction and release, depends strongly on the PIP2 subtype contained in the membranes of the donor and acceptor vesicles. So they produced a rank order in the donor membrane, and the major donor membrane is a 4-5 PIP2, and the major acceptor membrane is a 3-5 PIP2. So this opened, of course, as you might imagine, a large number of questions. So how does STAR-D4 recognize donor and acceptor vesicles? How does it discriminate between the PIP2 subtypes in the membrane? And if the subtype discrimination determines whether the membrane embedded extracts or releases, how does it do that? And what in the membrane embedding triggers the translocation either into or out of the STAR-D4? So to answer these questions and describe quantitatively the molecular mechanisms, we obviously performed MD simulations of STAR-D4 embedded in membrane systems enriched with one each of the 3 experimentally tested negatively charged lipids, these that I mentioned before. Each trafficking function, the extraction by the apo-STAR-D4 and the release for holo-STAR-D4 from holo-STAR-D4 was simulated separately in each of the 3 membrane composition for a total for each of 1.7 milliseconds of simulations. This was necessary, as you will see. So the schematic representation is that there was a phase 1 in which we created an equilibrated STAR-D membrane system in all the membranes. So the embedding of STAR-D4 is a process. The phase 2 is to continue the simulation from the embedded configurations and identified features of a pre-release state just before there is a release in the holo. There is everything I show you for the holo, there is an equivalent in the apo, where there is a pre-extraction state. And the aim 3 is to explore the process itself from the pre-release state and calculate the energetics of this release. And we did calculate the energetics with steered molecular dynamics, which was then turned into PMF calculations, potential of mean force, that allowed us to estimate the barriers of the creation of the pre-release state on a 3,5 membrane and the creation of a pre-release state on a 4,5 membrane. And I remind you that 4,5 likes to have the STAR-D4 extract rather than release. And so you see the barriers are much higher. So the same schedule of MD simulations performed in the apo STAR-D4 to produce the equivalent information. So one of the most intriguing early observations from the simulations was that the N-terminal region indeed embeds in the membrane as the Maxwell group has established with some mutations. And these interactions include both the sheets and the helix. That is, both regions from here, where I showed you from the helix, but also regions from here, which I showed you before. And upon holo STAR-D4 embedding, we find that the main region of interaction becomes the beta-1 region. And you see here the nature of the interaction with the beta-1 region, which is down here. It makes the holo STAR-D4 tilt on the membrane. And so the question remains, so what is the role of the ones that were not used in the conserved basic residues that were not used in the upper part of helix 4? Because in the lower part of helix 4, of course, they interact. So our hypothesis was that these these more distal binding sites have another role. And the role that we found was somewhat difficult to explore, and that's why these enormously long simulations were needed. And I show you here on spherical coordinates that the organization of STAR-D4 on the different membranes differs significantly. And I'll show you, just to emphasize the major significant differences. You see, this region has 4% population of STAR-D4. This is an inclination region, and you see what it is. And it has almost zero presence on 4,5 membrane, but it has 40% presence on 3,5 membranes, and vice versa. As you see, this inclination is like this on 4,5 and almost missing on 3,5 membrane. That is the role of these residues here. They make the difference between the two inclination, because you see that here these upper regions are totally uninvolved, and here they're involved. This is the upper part of helix 4. They help this inclination. So what you see here is that when we got to the pre-release state, we realized that the pre-release state is a very rare occurrence. The pre-release state is a very rare occurrence with this inclination. And it is rare even on the membrane where the release is the main function. but it is almost non-existent in the others. So the pre-release orientation indicated in these red circles, it shows that the positions are extremely rare, and that, let me just take this away because I don't, sorry. It is extremely rare. It is less frequency observed on 4-5 and almost never observed on the PS, the phosphatidylserine types of membrane. So because this is a rare event, we took use of the first machine learning type of algorithm that no, it's not the first we've created, that we've used in this project to reveal the structural dynamics of the formation of this pre-release state. And I'm showing you in detail these ML and AI measures because we really are aiming to show what they can reveal and why we should partner with them. So these various algorithms, and I'll introduce them one by one, have revealed the following: that the structural dynamics of formation of the pre-release state, which is that inclined state that I showed you, is for what it is for the subtypes that we described. And that was done by using our rare events detection algorithm. Once we have identified the rare events, the different triggers for these kinds of orientations that are the response of the STAR-D4 to the detection of which PIP2s the membrane contains was identified by another analysis, the lipid interaction mode and landscape, which I will introduce. And finally, once we have the response of the STAR-D4 to the embedding in a specific type of negative lipid containing membrane, the question was, so what triggers the whole path from this recognition to, in this case, the release, and also the extraction in the case of APOL? And that was identified by the N-body information theory algorithms. So the resulting mechanistic interpretation of the experimentally known functions, which I showed you before, and the effects of mutations that relate to disease and dysfunction repair for this system is based on the involvement of specific structural elements of this allosteric pathway that was finally identified by NBIT. And they are quantified interactions that were revealed by all these methods. So this is how the story of this regulation of steroid trafficking actually becomes a story of the molecular story of STAR-D4 in terms of its structure on the membrane, its dynamic response, and the traffic mechanism determined by the membrane in terms of these three algorithms. And that's the heart of the story. Thus, the story is these AI/ML analyses. It was recently, very recently, published in the Journal of Molecular Biology, and I am going to use just parts of it, and more of it. It's a very long article, actually, because it goes into explicit detail, because my aim is to make these things as accessible as possible so we can understand and utilize the functional mechanism of steroid trafficking to understand and embrace the applications of these AI/ML. So the story of the AI/ML analysis begins with the rare events detection algorithm, as I mentioned. It starts from the information encoded in the MD simulation trajectories, of course, and this is where it is. And this red algorithm identifies the dynamics data, in the dynamics data, the sequence of events in which specific structural motifs move together, and we call them components, so I will mention components many times, to dominate the conformation of the protein at specific times. So these are the major stops in the conformational change that leads to the function eventually. So these are in this lingo macro states of the dynamics. And for this, we used at the heart of RED is an algorithm called NMF, non-negative matrix factorization, NMF, which was described, and its embedding in the RED algorithm was described in this paper in 2021. And the machine learning algorithm is actually one that is now experiencing a new growth, and we fished it out from a Nature paper in 1999. And it starts, as you might remember from the beginnings of AI, with analyzing pictures, really images, and classifying them. So this paper illustrates the novel NMF algorithm and as it is applied to image decomposition and then reconstruction. And these are the components, the story in that paper. 50 images were presented to the algorithm to learn the elements of the system, that is, to learn what the system is composed of. Now, the beauty of Sebastian is an extraordinarily intelligent person with whom we still collaborate. And we have started after we discovered these collaborations that continue to learn the elements of the system. But the elements are not pixels. They are something else. The algorithm learned a representation of the picture that it transformed into a sparse matrix of spatially identified elements, that is, groups of pixels, if you want, in the image, that are found in the entire set of images. That is, regardless of which image you had, as you know, if you show an image and it's changed in this place and this place, then the pixels don't remain the same, but groups of pixels reoccur. And so these groups, as clusters of pixels with darkness differences, constitute the input to the AI algorithm. So this input to the AI algorithm identifies region composed of these input elements that are found in all 50 images. And they have different weights because if in the different images they appear a very large number of times, then they have a higher weight than if they appear a very small number of times. And that tells you that this is not interesting information. So the algorithm, the multiplication, and you will see what form they take, the multiplications of these two elements in the input produce a weighted component reconstruction of the original image. That means it organizes the images by what components had the highest weight. So now we want to apply it to trajectories. So what could be the input? In the original application, the input was obtained by training, from training by examining the images. So it fed the images, it found this sparse matrix. In RED, which is our algorithm, the MD simulation did the training. Because if there are groups of residues that cling together for a long time, that is equivalent to one of these dark spots. And so the dynamics produce structural frames over time, which are the equivalent of the images, if you want. And in these images, we need to recognize groups that go together and are persistent for a while, right? So this is what the contact matrix tells us. If we create a special contact matrix where at elements, residues, for example, that are at a given distance are considered like the black pixels that come together, then you get a sparse matrix that contains all the information in this set of pictures exactly the same as in the NMF. So the contact matrix is a sparse matrix and serves exactly the same function. What does this mean? That the time-ordered contact matrix, that is each frame in the trajectory, in an MD trajectory, has a corresponding contact matrix. And the time-ordered series of contact matrix contains the complete information about the behavior of the protein structure in time. So the contacts that change simultaneously in several consecutive frames indicate groups of residues that create a new conformational state. And it dominates the protein structure for the time corresponding to this group of frames being together. And in red, these groups are then structural components, and their contributions are the weights. You remember it was important to know the weights of these components. So this corresponds in the image analysis to this matrix that you see below. And the weights are essentially the weights, just as weights were used in the original NMF. So structural components are image components, so therefore we can put them together in the same way. And how does NMF do it? It decomposes the input into a spatial array and a temporal array, as shown here, and the multiplication of the spatial array, which is composed, which you see is a tensor of residues in 2 dimensions and components in 1 dimension, and the temporal matrix. The resulting data array is a record of the time-resolved structural rearrangement events detected in the MD trajectory. So the rare events detection methods transforms the information encoded into a trajectory into a series of function-related conformational states, while revealing the time at which each specific state dominates. So how do we interpret this? So it represents a time-ordered series of rare events of conformational change. Well, that is easy to represent. As you see here, here you see the components of the weights. So you see that the weights of the components, sorry. And you see here that each component, let's take the purple components, its weight increases. That means that it dominates the conformation for a while. And then as it starts going down because another component begins to dominate the conformation, and we are going into another macro state or a temporal conformational change in the structure of the molecule. And so the next step is to interpret these changes that we have recorded in terms of actual structural components. And as I showed you, component 4 dominates the conformation at frame 40, and that's B. And then C, in C, the next component begins to take over. And in D, it dominates the structure. So what you would expect for the trajectory in the whole of star dynamics is a representation like this, but we found an even more interesting representation. It is exactly the same thing. These are components, and as you can see, they go up and they begin to wane, and the blue component begins to go up as the red wanes, and then the red goes up. But there is one interesting thing that is of great importance in protein design, but not in the analysis of this mechanism. And that is that just as in this representation shown in the yellow scheme here, and in this representation shown for components 0 and 1, these are components that in combination are maintain their high weights throughout the trajectory. That is, they don't undergo the conformational changes that relate to function. Rather, they contain the residues of the structural core. So already we have a segregation between components, which you remember as our groups of residues, which promote function, and components that represent structure. You don't want to mutate some components that are constant there because you will not get the protein to fold, for example. So there is extremely important information for protein engineering. So what happens at these time points in the MD trajectory of star D4, the holo star D4, which we're interested in? So let's take the first point that you see up here, right? This one here. So in this first time point, the components, when we examine them, are residues. If you remember, I told you about helix 4, these residues on helix 4, and these residues on beta 1. And you see that these residues create a strong interaction inside the star D4. And the two structural elements come together. And here down below is an omega loop, and all its residues in this loop are parts of a component, and they interact strongly with this N terminus of helix 4. When component B appears, you see this is the new place of component B growing, then two things happen. These residues separate from one another, and these residues come together close, as you see, they come close by, and these residues separate. And that you can see here. The omega to H4 distance, you see in this blue, increases in the trajectory, and the beta 1 to H4 distance decreases in the trajectory. We followed them because that's what red told us. But something appeared that we had not identified as a component because we didn't use the cholesterol in the binding site as an element of the trajectory. We only analyze the trajectory of the protein. But look what happens. In the first component, the cholesterol is well embedded in its binding site. In the second component, you see in this time, you see what happens. The distance between the tail of the cholesterol and the beginning of the membrane, right, which is here, decreases significantly from this by 5 angstroms. So you can see that these components, the transition between these two components, are something that relates to getting to the pre-release state. And interestingly, down here, when the distance becomes 0, this represents the rapid release of the cholesterol in the membrane. So how did the holo star D4 recognize this as a target for cholesterol release? Well, I told you that one of the findings at the very beginning, I told you that extraction versus release depends on different binding modes of negatively charged lipids. And I want to show you how we discovered that. What you see here is that there is, and I will go to this in more detail, on this membrane, the 3,5 membrane on which this happens and release occurs, there is a PIP2 binding right at the heart in the upper part. I told you that we don't know exactly how the upper part of the H4 plays a role. There is a binding of this of the PIP2 from the membrane. Of course, this is a PIP2 3,5. Now, what you see here in the trajectory following the positioning of this, you see here that the PIP2, the distance of PIP2 to these residues, right? That's what we are showing here, decreases to 0. That is, the PIP2 comes and binds here. It begins to do so exactly at the same time in the first component, and it ends doing so exactly at the same time in the blue component, component number 3. So obviously, this is a triggering event in the release. So we need to discover how different PIP2 subtypes from the targeted membrane bind to the basic residues. And they bind obviously to the regions that have basic residues. There is a left group here. There is an upper group here, which I already told you does bind the PIP2. And there is a right group here. And to identify what the preference is, we use the PIP2 interaction mode landscape analysis. I won't go into detail of this. I'll just tell you what the algorithm does. It discovers the different negatively charged lipids from the membrane having different binding modes for STAR-D4. And it quantifies that. So there are minor modes of the PIP2 binding to STAR-D4, and I won't go into this because they're 3% and 12%. And there are major modes that this algorithm identifies for us. And what you see here is that the upper group is the most populated mode in this trajectory, and it has different orientations for binding. So we had to build a deep neural network to classify these binding modes, even in the major binding mode. And this was successful, and it predicted the subtype binding mode in this position correctly, as you can see, without real. This is the matrix, the resulting matrix from the DNN, and it reveals the molecular determinants, that is the specific interaction responsible for the PIP2 subtypes. Because if you look at which residues identified were important for classification, you identify them, and we identify them in this way. So here are two ML algorithms that allowed us to show that PI35 binds preferentially in the holo STAR-D4 in this region. The occurrence is 53% for 3,5 and only 3% for 4,5. So it's a selective region for 3,5. And the corner binding mode, the other binding mode, is selective for the 4,5 in exactly the same kind of selectivity. So this event is the recognition event on which membrane have I embedded myself, and it is responsible for uh, the target. So you see that our AI algorithms identify the evolution of the functional process in time and identify the conformational reorganization in the STAR-D4 function. And note that all these mechanistic steps are identified in terms of specific residues involved. So for example, we identified these residues here and these residues here and all the others, thanks to the algorithms used. So now we have a complete definition of the elements of the mechanism. But this does not yet represent a rigorous mechanistic relationship because if I'm telling you a story about the mechanism, I have to tell you, so you see, this happens first, and then this triggers that, and then this goes there, and then this happens at the end. All I'm telling you so far is that we've discovered that this is involved, and this is involved, and this is involved specifically by residue and by time at which it becomes involved. So now we need an algorithm that is a quantitative analysis framework capable of describing the dynamic connectivity path among these motifs that produces the complete mechanistic cycle of STAR-D4. And you see here a schematic representation of the dynamics if you want. And if you focus on the main place regions, the membrane PIP2 subtype recognition and binding, which is this region here, the cholesterol cargo binding site, about which I told you very little. It's interesting for design, but to make another transporter, but you understand that we have all those details. And the gate residues, which are involved in the changes down here, right? So we deliver a dynamic connectivity path. We will deliver among the structural elements of the structural motifs involved in the dynamic mechanism. And that is, for example, these three. And the transition among conformations that produce the metastable states in the mechanistic models will, because they come from trajectories, reflect energy-based behavior of the molecular system, because they are encoded in MD simulation trajectories. But I note for you that if you have sequences of NMR-based changes or crystallography sequences, that is exactly the same type of input for this particular algorithm. What it's not appropriate to do is to invent conformational states. That is, you say, well, I think this goes here and this goes here, so how would this be connected? That doesn't work for this rigorous algorithm. So for the STAR-D4 system, the resulting dynamic model will produce a consistent manner of the full cycle of membrane target recognition and functional performance of cholesterol uptake or delivery. And that algorithm was described in this PLOS computational biology paper, shown here, and then elaborated in great way in chemical reviews on an enormously long paper, which I usually try to do with algorithms that I would like very much for people to consider using instead of a patchwork of things. So NBIT uses, how does it work? It uses the formalism of Shannon's information theory to determine how intramolecular signaling is generated by actual communication that we learn from the trajectory between dynamically interconnected structural motifs. And the communication probability and pathways between these specific groups of residues are determined quantitatively from the trajectory. So this is a rigorous mathematical theory developed by Shannon, a formal framework that evaluates the connectivity between dynamics of spatially separated structural motifs. So If motif X moves and motif Y moves, and obviously the binding and the gate move, if there is encoded in the trajectory a connectivity, a structural information sharing, then the configurational entropy calculated from the covariance matrix in the MD trajectory, as shown in the paper, is essential because is an essential component of evaluating the mutual information term between X and Y. So I know that many people in the society and in our field have used mutual information, but I will just show you how mutual information is calculated directly from these entropy terms. But I want to show you that the calculation depends on a term that is the entropy of X conditioned on Y. So because it depends on the entropy of X conditioned on Y as discovered from as encoded in the trajectory, it answers a very interesting question, namely, how much does knowing the value of Y decreases the uncertainty about X? So if you imagine two things that move, I hope you see my fingers, two things that move like this, but every time, and they move erratically, but every time this is down, this is up. And every time this is up, this is down. That means that there is some information about the movement that I can glean about the movement of this finger by looking that I can get by looking at this finger, because I know for certain that if this is up, this will be down. I don't know anything else, but I know this is up, this must be down. So that is the power of the mutual information. So the n-body information theory goes beyond the pairwise mutual information formalism. So you don't need to construct a network and look at pairs because the network construction is arbitrary. To reveal a real communication path. So what do we achieve there is that the dynamic information about this PIP2 binding motif that I introduced before with this motif that I showed you is connected in the beta 1, and of this motif with the one that I showed you before is connected with the opening of the gate, you see, between H4 and omega 1, and that this motif shares with the binding of cholesterol, which prepares for release, and the performing of the release, which is the cholesterol release. That is the allosteric communication path that the n-bit produces. This is not an observation. It is quantified formally in the n-bit calculation of the coordination information. So if we take these three positions that I told you are of importance, we can calculate these terms, and they're very explicitly, carefully, and slowly described in the two papers that I mentioned, together with the total correlation. It describes the total amount of information that is shared among multivariables, that is, this group and this group and this group, in a whole set, which is the set of components of the functional description that the components that we achieved by the application of the red algorithm, right? So this expansion formalizes the connectivity among all functional residue groups, the ones identified from red, by co-information, which the property of which is that if the receiver is connected to this group and the transmitter is also connected to this group, then we know that the receiver and transmitter are connected. These names are not my invention. These are Shannon's invention because, as you might imagine, he talked about telephone communication. But that is the information theory. And then the mutual coordination information gives us channels beyond those, as you might imagine. So both types of channels illustrate the creation of indirect allosteric interactions, right? Because this is an indirect interaction due to this, both being connected to one. Together, this quantifies the flow of information that reveals long-range coordination of dynamic behavior of the kind that I showed you schematically before. So how do we translate quantitatively the schematic representation of connectivity? We connect it by using these channels, and that gives us a full matrix of transmitter-receiver, and we identify how this first event of binding of the PIP2 here, that is the PIP2 binding mode, what it connects most strongly. And interestingly, it connects most strongly with beta 1, which is what I showed you, and with the sites of the cholesterol binding site. So this is what starts the evolution of the pre-release state. And the rest is the extent to which these connections participate in turn, as described down here. So this is a quantitative representation of this connectivity. The PIP2 binding mode is the starting point that leads to beta 1, which I showed you is connected to H4 in the red algorithm, which connects to the other betas and therefore connects to the cholesterol site. So now the various algorithms have identified the structural dynamics of the formation of the pre-release state by detecting the components in the red. The different triggers, the triggers are different for the star D4 holo from the star D4 apob because the mode of binding is different, and that is what the lipid interaction landscape showed us. And finally, the allosteric pathways that was quantified by our N-body information theory. So the result is a mechanistic information. And everything you may want to understand about function, effects of mutations related to disease or dysfunction is based on the involvement now of specific structural elements of the allosteric pathways in a quantitative manner revealed by these AI/ML analysis. So if we contemplate the role of our collective work as QCMS members, it seems that making a partner of this enormous promise, capability, and intelligence of the right AI/ML approaches will help us achieve the scientific goals that we share. But we need to understand, adapt, and integrate these approaches in a manner that will complement and strengthen the physics, chemistry, and biology, which must continue to constitute a foundation without which our work cannot continue to thrive. And this is really what I wanted to leave you with. Thank you.